Thank you for the kind introduction. Yeah, I was going to say that my legs are shaking because it's cold, but I guess that excuse is gone now, so that's okay. But my name is Nikhil Milind. As I was introduced, I'm an MPhil candidate, and I'm studying Alzheimer's disease, not here, but in the past. And I kind of want to tell you guys about the challenge of complex diseases, which Alzheimer's is one of. So complex diseases actually have a very specific definition in genetics. But here are a few that you might have heard of, right? I'm going to use Alzheimer's as an example, but I want you to know that the analytical techniques I use here and that we use in genetics kind of apply to all of these diseases. And so there's always a genetic component. We generally do twin studies, sibling studies, things of that nature. And so Alzheimer's is a disease that we know is heritable. There tends to also be an environmental effect, right? So there have been hypotheses about metals and viruses affecting these different diseases, but those are mostly environmental exposures. There's also lifestyle. So there's a recent link between, for example, the Western diet and Alzheimer's, right? So all these three factors kind of confluence to produce a complex disease. When we think about them genetically, complex diseases are often compared to Mendelian diseases or diseases that we can track the inheritance of. So a classic example is sickle cell, right, where you have a mutation in one specific location in your DNA that results in one change in a protein, and that causes the phenotype that we observe. Complex diseases are more complex, right? So generally, you have multiple locations in the genome that start to matter. They tend to have small effects on the disease. Rather than affecting just one protein, they start affecting the entire complement of molecular machinery in your tissues. And they all add up to what we consider a complex disease. So that was kind of a broad introduction to complex diseases, but I've specifically worked in Alzheimer's disease, which is a progressive neurodegenerative disorder. The current estimates are about 10 to 30% of prevalence in people of ages 65 and older. And there are two forms. So the early form is actually very well studied. It is considered mostly Mendelian. On the other hand, the late onset form is what we traditionally think of as Alzheimer's disease. And it's sporadic. We know it has a genetic component, but it's not something you can track through families, for example. And there are no curative therapies for this disorder. So on the right here, I want to impress you with the idea that there are a lot of dysregulated pathways underlying Alzheimer's, right? The first kind of years of research went on focusing on amyloid beta plaques, which is kind of the main thing you see in Alzheimer's brain. But now we have come to appreciate that there's many different pathways that underlie this disorder. So let's say you're all geneticists, right? A fun role. And so you're thinking, well, I want to figure out which locations in the genome are associated with Alzheimer's disease, right? Why are you interested in that question? Well, for one, if you, in the future, we're moving towards precision medicine. I know that word was thrown out a few presentations ago, right? And it is super important, right? Part of it is if you know which genetic locations are associated with disease, you can assign a risk to a patient, and then you can treat them specifically based on their genetic makeup. In addition, understanding where in your genome is associated with the disease tells you something about how you might be able to cure it, what mechanisms underlie the disease, right? So you're a geneticist, you start going across the genome, and you look at places where people have different variants. And you bin people by their variants. So you're seeing here three baskets, right? People with genotype GG, genotype GC, and genotype CC. And you can see that Alzheimer's disease here and elderly controls are not really associated, right? So you can separate people by their genotypes, and there seems to be no association. Other variants, you might actually see some association, right? So here, people with the TT genotype seem to have not protection, but they have lower incidence of Alzheimer's disease, and then people in the GG bucket have a high incidence. And so you do this scan and get this type of plot. This is called a Manhattan plot. Each point here represents one of those variants that I showed previously, and the height represents the significance of that association. Again, this is not causality, right? We always hear correlation is not causation, but again, these are associated variants. And I apologize for this busy plot. This is actually not part of my research. This is from a study from 2020 that is yet to be published. But I want you to focus on this variant, or this gene called TMA106B that has very recently in this study been associated with Alzheimer's disease. So this is all to motivate the fact that these researchers required 450,000 samples to discover the signal. I'm going to show you how our lab did it with 600 patients. Right? So the way they performed their study was to compare associated complex disease directly with the genome. The idea in our lab was, well, let's add the transcriptomic layer to this model and try to add layers of mechanistic information to our analysis. And so that's kind of what we did. So on the figure on the left there, I'm showing you some of the pathways we discovered from our transcriptomic analysis. You'll notice that many of them were similar to the ones that I showed in that introductory slide. And in fact, we were able to stratify our patients with Alzheimer's disease into 2 broad clusters. And you'll notice that compared to controls, there's actually a lot of heterogeneity in these pathways, right? Which is very surprising, because if all of them have the same disease, you would expect that dysregulation of those pathways also aligns against controls in the same direction, right? But that doesn't seem to be the case. And we were wondering, well, if there's heterogeneity, there might be a genetic component. So similar to what we do with case control studies, we can walk along the genome, score patients based on their association with these pathways, and perform an association study, right? And so a similar idea, if we see something like this, that means that genetic location is associated with that pathway. And the results look very similar. This is part of my research. And want to point out that, the laser works, okay. That TMEM106B actually pops up in this analysis associated with oligodendrocytes and neuronal activity. And if you actually separate patients by subtype, right, subtype A and subtype B that I showed previously, subtype B has the signal present while subtype A does not. And so the important story here is that we have added layers of mechanistic information that are not present in the case control study. We've shown that there are specific cell types that are associated with this variant. We can show that there's a subtype of patients where this variant matters and a subtype where it doesn't. In fact, subtype A has upregulated inflammatory markers and subtype B does not, which is very surprising because in Alzheimer's disease, neuroinflammation is considered the hallmark late postmortem pathology. And so it's very surprising that there's a set of patients that develop the disease and don't have have reduced neuroinflammation compared to controls. What's very interesting there is that TMEM106B has been implicated in a neuroprotective function. So this variant actually protects against inflammation. But since those patients are still developing Alzheimer's disease, we know that there's some pathways before the neuroinflammation occurs that is likely causing Alzheimer's disease. So my conclusion here is twofold, right? First thing is that case control studies required a huge amount of individuals across a huge cohort. Our transcriptomic stratification only required 600 people to get the same types of signals. And this is a broad form of study called multi-omics studies, right, where you add a lot of molecular information to study genetics. And so I just want to impress the idea that in addition to associating variants, we are able to add mechanistic context and also increase our resolution in this disease. So I'd like to thank, of course, the people in my lab and the consortia. Thank you, and I'll take any questions.
Thank you very much for this talk. I know you wanted to try the black button. I know. I saw it on you in the beginning. Anyhow, oh, wonderful. I don't have to ask either.
Could you explain, please, what the transcriptome was here? Because I don't think I've understood. It was a great talk, by the way. Thank you very much, but I just...
Yeah, no, absolutely. I think that's a great question. So the ohms, right, the multi-omics, the ohm is kind of like a new buzzword, if that makes sense. It all started with the genome, right, and then people just added on to it. So you guys might have heard of mRNA recently, right, because Moderna, for example, is an mRNA-based vaccine, right? And so generally what we call the central dogma of molecular biology states that, you go from a gene to an mRNA transcript to protein. That's kind of how information flows through from genes to function. And so the transcriptome is the full complement of mRNA that's present in a tissue, right? So if you take the mRNA from all the genes, right, it tells you if genes are active or not, right? So if a gene is very highly expressed, but you do that for all 20,000 genes in the human genome, it's a great question. And so the proteome is the same, metabolome, epigenome, all are kind of these ideas of high throughput assays, as we call them. Yeah.
Thank you. Any other questions? Wonderful. I'm doing my exercise. And going back.
Thank you. Do you know well, do you think there are ways to test, for example, whether this particular gene, you know, like experimentally, basically, if there is a causation type?
Absolutely.
And also,
yeah.
sorry, is it known what this protein is doing in like normal brain function?
Yes, yes. Absolutely great questions. As I cautioned, right, correlation is not causation, right? Yeah, we have made an association, but that does not mean much. In this case, TMM106B has been previously associated with frontotemporal lobe dementia, right? So we know it has a neuronal function. It's associated with lysosomal activity, specifically trafficking. The specific variant that we associated is, and this is getting slightly genetic-y, right? But it's associated with disrupting a CTCF binding site. So it changes the conformation of DNA in a local region of the genome. And that's what people believe somehow functionally results in an immunoprotective phenotype. But no, nothing has been done past that. There are mouse models that are currently being developed with this variant. Yeah, so we'll know soon. Yeah.
Thank you. I'm checking the time in the meantime. So we have time for one quick question, if anyone is volunteering. Yeah.
Do we have any more information about the difference between the type A and type B patients that you described earlier?
Yeah, that's a great question, right? So one issue is that when you look at this plot, it doesn't really look like two clusters, right? So it's really hard to say, you know, are we just identifying variation across the first principal component, or if they're like two real clusters? But once we've done this analysis, the biggest signal is inflammation, right? But so what that means is subtype A has patients that have a lot of brain inflammation compared to subtype B. But in addition, they actually differ in all those aspects. So all those pathways are different between those two subtypes compared to controls, which is very surprising. It's a very large amount of heterogeneity for a population that is supposed to be experiencing one disease. Right? But that's all we have, really.
Thank you very much for the
Thank you so much.
presentation again and for the